Hello, Bio 102. This is the lecture for Chapter 10, Photosynthesis. Because we didn't have a chance to have a lecture on March 24th, this is the online lecture. So, photosynthesis is the process that converts solar energy into chemical energy. And autotrophs are the organisms who carry out photosynthesis. So they're the ones who produce organic molecules. They take carbon dioxide and other inorganic molecules, non-living stuff, and they turn it into organic molecules. So they're able to sustain themselves without eating anything else. That's why they're called autotrophs, because they make their own food. So here are some pictures of some different autotrophs, including the plants, the algae, and some single-celled photosynthesizers. So, we already know that chloroplasts probably evolved from photosynthetic bacteria that were engulfed by other bacteria during the endosymbiont process that resulted in eukaryotes. The chloroplasts are green because they contain a pigment called chlorophyll. The chlorophyll is the pigment that absorbs light energy and actually drives the synthesis of organic molecules inside the chloroplast. So chlorophyll is really the molecule that's critical to photosynthesis. The chlorophyll is embedded of the chloroplast. Here you can see we're zooming in on the edge of a leaf, and you can see different kinds of cells, including the stomata embedded in the outside of the leaf there. And then if we zoom in on a cell called a mesophyll cell, we can see that it has these green compartments within it, and those are the chloroplasts. And the chloroplast has an outer membrane. It also has an inner membrane, so it's actually sort of 3 membranes instead of 2 like we saw in the mitochondria. But the important thing to note here is that it's got these outer membranes, and then it's got the thylakoid membranes, which divide the chloroplast into the stroma, which is the area outside of the thylakoids, and the thylakoid space, which is the area inside of the thylakoids. So, the equation for photosynthesis that we'll use in this class is the one that shows the net consumption of water. So, 6 CO2 plus 6 H2O give rise to C6H12O6, which you know is glucose, and 6 O2. It's important to remember that you have seen this exact chemical equation before, just with the arrow going the other way, because photosynthesis is the opposite of cellular respiration. In cellular respiration, we take glucose and add oxygen to get energy. That's energy that we use for cellular processes, and give off CO2 and H2O as waste products. Here in photosynthesis, CO2 and H2O are the materials that are needed to carry out photosynthesis, along with an input of energy which comes from light. And glucose is the product. It's what's being produced by photosynthesis. And then oxygen is given off as a waste. So we're going to focus on two different stages of photosynthesis, the light reactions and the Calvin cycle. The light reactions are the photo part of photosynthesis, and the Calvin cycle is the synthesis part. In the light reactions, which occurs in the thylakoids, water is split and oxygen is released, and NADP plus gets reduced to NADPH, and ATP is generated through a process called photophosphorylation. So we'll look in more detail at how each of these things works in just a minute. Meanwhile, in the Calvin cycle, which occurs in the stroma, remember that's the area outside of... First, here's a diagram showing that overview that I just described of the light reactions, the Calvin cycle, what comes in and what goes out. This is a diagram you should know. This is really important because it shows what's being used in photosynthesis, what's being produced by photosynthesis, and it shows those important connections between the light reactions and the Calvin cycle, where what's produced by the light reactions gets used up in the Calvin cycle, and vice versa. Okay, now we'll focus in on light. We know that light is a form of electromagnetic radiation and that all electromagnetic energy has waves. And the wavelength is the distance between the crests of a wave, and it determines what type of electromagnetic energy we're talking about. So we have this whole big electromagnetic spectrum, and visible light, the light that we can see, magnetic spectrum. So here you can see at the top the whole electromagnetic spectrum, including X-rays, ultraviolet radiation, infrared, microwaves, radio waves. Those are all types of electromagnetic radiation, but we're just going to zoom in here on the one little section of that whole spectrum that contains visible light. And then within that chunk, there's also a spectrum going from 380 nanometers up to 750 nanometers, representing the different colors of visible light. An absorption spectrum is just a graph that shows how a pigment absorbs light in different parts of the visible spectrum. So basically, at different wavelengths, how much light is... So here we can see the absorption spectrums of chlorophyll A, chlorophyll B, and the carotenoids. I want you to really focus on chlorophyll A, which is that kind of bluish green line. And you can see that most of the absorption is happening down in the violet and a little bit blue part of the spectrum, and way up in the red and slightly orange part of the spectrum. So green and yellow are basically not being absorbed by chlorophyll A, and that's why leaves look green to us, and that's also why green light is not very useful for carrying out photosynthesis. So what happens when this pigment, chlorophyll, is absorbing the light? What happens is that the light actually bumps an electron from the ground state to an excited state. Remember way back at the beginning of the fall semester when we learned about electrons and how they're in their orbits and they tend to fall into the... Here's a kind of silly but I think really useful diagram showing how those photons of light are affecting the electrons and then how those electrons, those excited electrons, are being used to do what the light reactions need to do for photosynthesis. So over on the left-hand side there you can see one photon of light comes in, providing energy and bumping that electron up to an excited state. That's on top of the first tower there. That's photosystem 2. Don't worry too much about photosystem 2 versus photosystem 1, but you should know that the electron gets excited, and then as it falls back down, it's used to pump protons across a membrane for chemiosmosis. We've seen that before, and we'll see it again in a minute. That's the process that produces ATP. But before that electron can fall all the way back down again, another photon of light bumps it up even higher on the top of the second tower there, where it can be used to reduce NADP plus to NADPH. And you can see, that provides the two outcomes of the light reactions. ATP, which is a source of energy, and NADPH, which is a source of reducing power. So remember, we learned about chemiosmosis once before when we did cellular respiration as part of metabolism, and we learned about how it happens in the mitochondria. Now we're going to see that the way it works in chloroplasts is actually very similar. The spatial organization of both of these two, the chloroplast and the mitochondria, is similar in that a membrane divides the space into two different areas. So in mitochondria, protons are pumped across the membrane from the intermembrane into the intermembrane space, and then as they diffuse back into the mitochondrial matrix, they drive the synthesis of ATP. So I told you that there are a bunch of different diagrams in the book that show how this happens. This is the simplest one, and this is the one that I'd like you to focus on. I also think it's really helpful because it shows how similar this process is in the mitochondria, which we've learned about already, and the chloroplasts, which we're learning about now. So in both cases, you have protons being pumped across a membrane. In the mitochondria, the energy for pumping those protons comes from the sugar that's been broken down. In the chloroplast, that energy comes from the sun. But in both cases, the proton is pumped across a membrane, and then as the protons diffuse back across the membrane, they go through ATP synthase, which we've seen before, and ATP synthase drives the production of ATP. So now we've produced ATP and NADPH, and those two components are going to be used by the Calvin cycle. So the Calvin cycle, like the citric acid cycle, regenerates its starting material. It starts out with a particular organic molecule, and then as it goes through the cycle, it regenerates that original organic molecule. The Calvin cycle is kind of the opposite of the citric acid cycle, though, because the citric acid cycle was breaking down sugar, and the Calvin cycle is building sugar. So the Calvin cycle is going to build sugar out of CO2 using energy from ATP and the reducing power of the NADPH. So carbon comes in as carbon dioxide from the atmosphere and leaves the Calvin cycle as a three carbon sugar called G3P. And so in order to make G3P, the Calvin cycle actually has to go around 3 times so that it's grabbing 3 molecules of CO2 and making 1 molecule of this 3 carbon sugar called G3P. Obviously, the Calvin cycle is going to need to happen twice G3P is going to need to be made twice in order to form glucose, which is a 6 carbon sugar and which is the stable outcome of photosynthesis. So those G3Ps are going to get glued together very quickly into glucose molecules. Now, one thing I do want you to know about the Calvin cycle is that the original enzyme, the enzyme that actually grabs the CO2 and brings it into the Calvin cycle, is called rubisco. Rubisco is short for ribulose bisphosphate carboxylase oxygenase, but you don't need to know that. You can just call it rubisco. And rubisco is a really important protein, and it's going to be really important in some of the last slide here is just a picture of the Calvin cycle. And just like with the citric acid cycle, I don't expect you to memorize all of these intermediate compounds. Thank goodness. Basically, what I want you to know is CO2 comes in at the top up there and gets fixed by rubisco. You can see rubisco there shepherding the CO2 into the Calvin cycle. The output is a 3 carbon sugar called G3P, and you can see that the original starting component of the Calvin cycle, which is ribulose bisphosphate, is regenerated. That's what makes it a cycle. You can also see on the edges there ATP going in, being transformed from the high energy ATP to the low energy ADP, and you can see NADPH going in and being transformed from its reducing form back to the oxygenated form of NADP+. So you don't need to know all these details, but you do need to know what goes in and what comes out, and the fact that the starting material gets regenerated.